Hello, good morning. Today we'll have the concept of B cells and T cells. B cells are very important lymphocytes which are responsible for humoral immunity. They proliferate and mature in the bone marrow. The B cells which do not recognize the host tissue as self are eliminated during the maturation process by undergoing apoptosis. This process is known as clonal deletion. After maturation, when they get the B cell receptor over them, they will leave the bone marrow and move to the lymphoid tissue, where they encounter various pathogens. Lymphoid tissues like spleen, lymph node, and marl. Now B cells, they work in two ways. B cell, if they recognize the antigen, then they are activated, they proliferate, they produce huge population of plasma cell and mammary cell. This process is known as clonal selection, and they will then produce antibodies. Mammary cells, they are for future use. Plasma cells, they produce the antibodies in large amount, which then bind with the antigen in the plasma through neutralization, through precipitation, through complement fixation, and nullify the antigen. Some B cells are activated when the antigen binds on the receptor and stimulatory signals, they are generated inside the B cell. The B cell, it will then process the antigen, and helper T cells, they are required. If helper T cells, they are not present, such antigens, they are not showing any response. So such a type of antigens, they are known as T dependent antigens. When the T cells, they will join the B cell, they will produce cytokines, and these cytokines, they stimulate the mammary B cells as well as the plasma B cells to produce huge amount of antibodies, which will then work to show the humoral immunity. So it is very clear that humoral immunity is the immunity which is provided through the fluid, that is the plasma, the proteins which are present in the plasma, and the proteins which are present in the plasma which are showing the humoral immunity, they are the antibodies. So either directly the B cells, they'll produce antibody, or bind with the T cell and then they produce the antibody. Then they will neutralize, they will lyse the cell, and ultimately the component which is formed, that is the antigen antibody complex, that should be removed. Otherwise it will clog the capillaries and it can cause problems. So through phagocytosis, that complex is removed. Now see here the various functions of the antibodies. Antibodies, they are responsible for inducing opsonization, activating the complement fixation, then removing the neutralization of viruses as well as the toxins is done. Then antibody dependent cytotoxicity, it is performed. Then direct antimicrobial activity, directly they can bind with the microbes. Then reduce the damage to the host from the inflammatory responses and the generation of the adapters. Even immunomodulation is also done. Now, how many types of antibodies they are produced? Through this process, 5 different types of antibodies they are produced: IgG, IgE, IgD, IgM, and IgA. Basically, IgG antibodies, they are about 80%, and they are responsible for resistance against viruses, bacteria, and bacterial toxin. IgE is produced in very small amount and is responsible for the allergic reaction. and it is present on the surface of basophils and mast cell. IgD is also present on the B cell, and it is responsible for T-dependent antibody production. IgM is a very important antibody which is pentamer, and it is responsible for binding with the antigen in huge amount, and even the blood types, the agglutination, it is because of the IgM. IgA is secretory antibody which is present in the mucus, tears, saliva or the semen, and it is responsible for binding with the antigen. Now the second type of cell is T cells. T cells, they are proliferating in the bone marrow and they migrate to thymus for maturation. That is why they are known as T cells, because they mature in the thymus. Now the immature T cells in the thymus who does not recognize the self cells are deleted by apoptosis, and again this process is known as clonal deletion. After maturation, the T cells, they will leave the thymus and move to the lymphoid tissues, that is the spleen, that is the lymph node, that is the malt, where they encounter with the pathogen and are responsible for cellular immunity. Now the maturation takes place in the thymus as the thymocytes moves from cortex to the medulla, and then they are released into the bloodstream. The maturation of T cells leads to the formation of two important types of cells, that is cytotoxic T cells and helper T cells. Cytotoxic T cells, they are with CD8 present on their surface, whereas the helper T cells, they have CD4 present on their surface. Very important, the T cells, they will have T cell receptor, they have the CD3 and CD4 or CD8 depending upon the type of the T cells. Now here again, those who does not recognize the self cells, they are eliminated by apoptosis and then bloodstream. Now helper T cells and killer T cells, they work in different way. Helper T cells. Helper T cells, they require antigen presenting cells. Antigen presenting cells, they are macrophages, B cells, dendritic cells. They will bring the pathogen, they will process the pathogen, and the pathogen will come on the surface of MHC class 2 molecule. So helper T cell binds with MHC class 2 molecule. We can say that T cell receptor can bind only with the processed antigen, not directly with the antigen present, right? So T cells, helper T cells bind with the MHC class 2 and they will lead to the production of interleukins. Interleukins or the cytokines, which then stimulates macrophages for phagocytosis, stimulate CD8 T cells for lysis, stimulate B cells for production of huge amount of antibodies, as well as activating other CD4 T cells. So we can say that CD4 or the helper T cells, they will work through cytokines. Then coming to the second is the cytotoxic T cells. Cytotoxic T cells will bind with the cells, basically the nucleated cells having MHC class 1. So the pathogen, it will enter here and then it is processed. The processed peptide, it will be presented on the MHC class 1 molecule. MHC class 1 bind with TCR, and then what happens? The TCR, it will produce perforins. These perforins, they will create hole in the pathogen and ultimately it will kill that. So helper T cell work through cytokines, whereas cytotoxic T cells works through perforin directly attacking the cell. So we can say that the helper T cells, they perform in two ways through cytokines. MHC class 2 molecule bind with the helper T cells. They will release the cytokines. They will be responsible for activating the cytotoxic T cells. They will be responsible for activating the B cells for the production of antibodies, as well as self-activation of the helper T cell is done by them. See here, the cytotoxic T cells, they are having large number of granules inside them, basically the perforins. They will bind with the MHC class 1 molecule. They will release the enzymes. They will make hole on the surface of enzyme. So cell is dying with the help of apoptosis. So in this way, the cytotoxic T cells, they will kill the pathogen. I hope the concept is clear.